So, dear Chair, dear colleagues, thank you very much for the kind introduction. It's really a pleasure and an honor for me to be here and to discuss this very interesting topic with you. Before I start, I have to state a conflict of interest. It's only a theoretical one, but I want to let you know that I was involved in the development of a ready-to-use calcium ionophore. So I have no financial interest, but all the studies that we did and that we did publish are based on a ready-to-use ionophore solution, and we got this product for free, so we didn't have to pay for it, just to let you know. At first glance, if you look on the topic that I've been given today, you'll see that, or you might think that total fertilization failure, and on the other hand, impaired embryological development in case you already had a fertilization, are two completely different things. But I hope at the end of the talk, you may be convinced that it's all more or less a matter of calcium deficiency. So if the calcium is okay in your cell, you might succeed and you may have a transfer. Of course, I'm not talking on apoptosis and senescence. These are all other reasons that an embryo might stop. I'm just talking on the calcium level of the cell. The outline of my talk today is that I'd like to start with some principles of oocyte activation, and then I try to convince you that it's important to distinguish between having a problem and an accidental event. So you have to be sure that it's really a failure of fertilization or of embryonic arrest. So that's quite important, I guess. And then, which is most probably most important for you, I'm gonna talk on 3 treatment options that you may have as a solution. And of course, nowadays we have to talk on safety issues as well, and there will be at the end some future aspects with this methodology. By definition, oocyte activation is one of 6 major steps of in vivo and in vitro fertilization. We know that the first 3 steps are bypassed by ICSI, but once the sperm is in the oocyte, you have to face oocyte activation, sperm processing, and PN formation as well. It is, by definition, also a physiological process that is composed of 2 events, which is the Sonner reaction that happens first on one hand, and on the other hand, it's the resumption of meiosis. It is generally acknowledged that the information to start these processes stems from the sperm. In particular, there are a couple of sperm proteins that may be are relevant for this start of the processes. Amongst others, there are 2 likely candidates, which is the post-acrosomal WW domain binding protein, and the best researched agent in this list is phospholipase C-SEDA. I'd like to go a little bit into detail. So if the sperm fuses, or the sperm membrane fuses with the oocyte membrane, or if the ICSI brings the sperm directly into the ooplasm, then this sperm protein, PLC-SEDA, is released, and it causes one important thing. It cleaves a molecule that is called IP2, or phosphatidylinositol biphosphate, which is PIP2. So this is cleaved into 2 molecules. One molecule is used for the Sonner reaction. This is diacylglycerol, and the other one is important for oocyte activation, and this is inositol 3-phosphate. So this inositol 3-phosphate, or IP3, goes to its receptors, and the receptors are located on the membrane of the endoplasmic reticulum, and this causes the release of calcium into the cell. So it's quite a complicated step until you get the calcium, and the calcium is presented in the form of oscillation. So it's not an efflux, it's always stopped, and once the calcium is in the cell and it's enough, it stops, and this has something to do with the sensitivity of the receptors, and then the storages are refilled again, and then the calcium again is released. So it's in the form of oscillations. And these oscillations, they stimulate mitochondria and produce ATP. So they produce their own energy. And you may see, and you might know, that the calcium signal of every single cell is different, and it can be measured. It's a matter of latency, of amplitude, of the duration of a peak, and of the frequency of those spikes. And most importantly, the error under the curve reflects the total amount of calcium that is free. And coming back to the main topic of this presentation, complete fertilization failure. If you look here on the right side, you see if you have no fertilization in a couple and you analyze the sperm, you bring the sperm, for example, in mouse oocytes, you won't get calcium peaks. So there is no oocyte activation potential in these sperm. On the other hand, we have a quite normal fertilization rate, and you see numerous peaks. And the effectiveness of this calcium signal depends on the number, the frequency, and all the other things I mentioned before, but also on methods like vitrification, in vitro maturation. Whatever you do with the cell might change the calcium oscillation pattern. And the competency to release calcium has to be acquired during maturation. So I found this nice image, and I have to thank my Belgian colleague. A prophase 1 oocyte gives you less and smaller amplitude calcium peaks as compared to mature oocyte. And during the maturation process, the oocyte acquires competency, it gets higher numbers of receptors, the sensitivity of these receptors is increased, it's a redistribution of the endoplasmatic reticulum to the sites where it's needed. So it's a lot of things that's going on in the oocyte. So now, how to diagnose that you have a real problem and that it's not happened accidentally? Because if you have only 1 or 2 oocytes, or you do a suboptimal ICSI with 5 oocytes, you have no fertilization, maybe you did something wrong, or it was a limited number of oocytes. But in case you have more than 3 oocytes, and you have no fertilization after regular ICSI, then you should at least consider to have a problem. And Belgian colleagues, they stated that it's very important to tell the patient whether the problem is with the oocyte or whether the problem is with the sperm. The good thing is, in IVF, whatever is the reason for the problem, you only have to increase the calcium. Because if there is no PLC setter in the sperm, you don't have fertilization. If there is no receptor for IP3 in the oocyte, you have no activation as well. So, but if you bring up the calcium, you might succeed. So they have a 3-step model. They do immunostaining of PLC setter in sperms to localize this protein in the sperm and to see whether it actually has it. They use buccal cells and analyze it genetically for the gene of PLC setter, and, which is most importantly, they bring in sperms of the patients, and we did it quite often. In case I don't succeed with any method, I send the sperms of the couple to Belgium in a frozen way. They inject the sperms into mouse oocytes, and they give me the results of their, what they called MOAD test, mouse oocyte activation test. If you have a look here, they have a negative control, they have a positive control of a known fertile donor, and then they bring in the patient's sperm. And what do they get? Either no activation at all or a very high activation. Of course, no activation does mean there is no potential of the sperm to fertilize an egg, a human egg. And on the other hand, if you have a high activation rate, then it's of good prognosis, and the problem lies with the egg. But there is this group in between 21 and 84% of activation. This has been considered to be quite normal, and actually it's rather a problem of the oocyte. But then this paper of Nomikos came out, and the Belgian colleagues, they realized that actually human PLC setter is more effective than mouse PLC setter, and they are not sure again that the MOA test is sensitive enough to detect these problems. So what they do is they make the MOCA test, the mouse oocyte calcium activity. That's what they measure. And they found that to be honest, the MOAT group 2, which they consider to be a normal one, is in fact a bad prognosis patient group. So only these activation patterns would be of good prognosis. And the good thing is they also found a threshold. So they counted and measured and analyzed all the data, and they found that the threshold is 9 peaks of calcium. If a human sperm creates at least 9 peaks in the oocyte, in the mouse oocyte, then the problem lies with the egg and rather not with the sperm. What they also found is that you should actually use a human oocyte, which they called HOCA. They would give you the best results. So now let's move on to the next step. How can you treat fertilization failure? The problem is, of course, it already happened. You can't, if you have no fertilization on day 1, it's too late for this cycle. So, but next time, if you would repeat ICSI without doing anything, I think this is kind of a major failure. At least you should try something, whether to do a genetic analysis or trying to activate it artificially. So the first paper I found, and I had the problem with this patient a couple of years ago, was a mechanical activation of the oocyte, a modified ICSI. And this has been published by Tesarik in conjoint collaboration with Laura and her group. And they did enter the oocyte with the needle, and then in a rather invasive way, they moved around the pipette trying to destroy the internal calcium storages. And indeed, it worked in oocyte-borne problems as well as in sperm-borne problems. And in addition, calcium from outside entered the cell as well. So in total, the calcium seemed to be sufficient to activate oocytes in these case series. But they did it at the expense of a 21% degeneration rate, which may not be a problem if you have 15 oocytes, but if you're dealing with 2 or 3 or 4 oocytes, 20% damage rate is too high. This led us to consider an alternative mechanical ICSI. And our idea, our hypothesis, based on 3 papers. One was the paper of Rainier and co-workers. He could distinguish between a group with fertilization and a group of no fertilization at all on the basis of the copy number of mitochondrial DNA. In other words, it's a deficiency in ATP, for example, that also caused, and of course it has something to do with calcium as well, which caused the no fertilization. The problem is we didn't know where these mitochondria are. But Jonathan van Blercom nicely published this paper and showed that the active metabolic mitochondria, they are in the periphery of the oocytes, but you can't see it. But this paper from Naples showed that the granular area, at least it suggested that the granular area contains metabolic active mitochondria. So what we did is to enter the oocyte with the glass pipette, to go to the opposite side and to aspirate only one time on the other edge of the oocyte, because this theoretically contains the metabolic active mitochondria. And then we went back and placed this cytoplasm in the center together with the sperm. And by doing so, we could treat 14 patients that were collected from all over Austria. So this was a conjoint study of the Austrian embryological group. So they had never fertilization and a transfer before. And with this slightly adapted Tesarik method, we found half of the oocytes fertilized and at least every third got pregnant. So in these years, we were quite happy. But nevertheless, we also had 8-9% of damaged oocytes, which is not acceptable. So we looked for other methods. The second method, and I will to skip this because this is more like a Frankenstein's lab, and I'm not sure if you have the gadgets in your lab to use electrical current voltage that opens the membranes and creates pores, and it has nothing to do with complete fertilization failure. That's the problem. But theoretically, if you have these instruments, you could do it. Most likely, you use or you might have heard of chemical activation approaches. This is most commonly used in the IVF world, and this is particularly funny because I told a couple of minutes on this oscillation. So in a physiological way, you should have oscillations. But ionophores, or most of these compounds, they only give you a single calcium transient. So there is a single peak, and then the calcium goes down. So this is not physiological at all. But it seems that the calcium, the area under the curve, is sufficient to cause oocyte activation. And two reasons may explain that. The first thing is that in literature, it could be shown that it's actually the first peak. The first peak is the most important one because it starts the whole process and all the downstream protein effects on calcium calmodulin dependent protein kinase 2, for example, which is the most researched protein that has something to do with oocyte activation. And this protein actually, and this is the second explanation, can act without calcium because it can autophosphorylate itself, and then it's active for a longer time. So you see, some of these compounds, they do create oscillations, but if they do so, they are less effective and limited to some species and not applied in the human, except strontium chloride. So now we have the real problem. So the good thing is that most of us use ionophores, either ionomycin, and I can state that this is more effective. Ionomycin, it has been shown in sea stars, is more effective than calcimycin. And recently, Nikiforaki in Fertility and Sterility compared the ready-to-use compound with ionomycin, but ionomycin plus injection of calcium chloride during ICSI, then ionomycin, half an hour rest, and another stimulus with ionomycin. So actually they had 3 stimuli, and it turned out to be more successful in terms of calcium creation than a ready-to-use compound or calcimycin alone. So this is the nice paper of Frauke van den Meerschout, and she showed, and this is the real problem, that there is not a single method in literature that tells us how to proceed. So what the authors did, and we are also included, so we use the ready-to-use product, but others use ionomycin. Others use a twofold versus a single application. The dosage it goes from 5 micromolar to up to 20 micromolar. So there is no uniform approach, and that's a problem, and that's why you can't compare all those studies. Coming back to the total fertilization failure, I'd like to draw your attention to the left side of this slide. So these actually are patients that had never a fertilization before. So this is the previous cycle, and this is the treatment cycle with calcimycin. And they ended up, and this is quite interesting, where is the fertilization rate? With some 40%. So all you can get with calcimycin is a fertilization rate between 40 and, in our hands, I think it's 55% of fertilization. Ionomycin gives you 70%, 60 to 70% of fertilization. But you have to mix it your own, and it's not mixable without the help of DMSO, for example, and it's not certified. So it's up to you to decide, and to the legislation of your country, whatever you do. But coming back to this talk, this was a retrospective study, and we did the same together with Markus Montag from Bonn in a prospective way in patients that had quite good sperm, so more than 5 million. So we thought the problem was related to the oocyte. And you see 47% fertilization rate in a group without fertilization at all. The same holds true for severe male factor. In these cases, the sperm is the problem because it was cryptozoospermic and azoospermic patients, and we saw half of the oocytes were fertilized. This case I will skip. This is a Cartagena syndrome. We also used ionophore, and we had a healthy live birth of twins, although the embryos don't look very healthy. So now, this is the very last part of the talk. Why does calcium ionophore help to stop embryo arrest, embryo delay, or reduce blastocyst formation rate? And this is a very interesting model from Lahmann, and he showed, and in green you see the PLC zeta. So you have the spikes that I was talking about, and suddenly, all of a sudden, during the formation of the prenuclei, the oscillation stopped. So it is thought that the PLC zeta is located now in the prenuclei, and after interphase, once the membranes disappear, then PLC zeta is released again, and then you have single spikes or fewer spikes prior to every mitotic division. So that's why we used calcium. And this is also interesting. In arrested human embryos, there is no calcium at all. So this has been proven by a Portuguese group. So we used an ionophore trying to save these embryonic arrest problems. But before, I'd like to show you this case report from Egypt. They had a patient where all the zygotes, they were fertilized but never cleaved for several subsequent cycles. And then they used the ionophore, and then suddenly it resulted in pregnancies. So this is the study of our group, published in Human Reproduction. So we focused on complete arrest, on complete developmental delay, so 24 hours at least behind in development, and reduced blastocyst formation rate. And what we found is that the survival rate until the day of transfer, which was 4 or 5, was significantly higher, and the blastocyst formation was also higher in all 3 groups. So somehow calcium went up in the cell, obviously, and the cleavage was a little bit better as compared to the others. And we had a quite good live birth rate of 45% in this particular bad prognosis group, I would say. So, safety issues. You might be aware of the discussion that's going on, and you will see it in the current trends that you have in your bag. Some authors claim that it's not sure. Of course, calcium ionophore usage is not physiological. It should only be used if it's the right indication. And if you think that it might be of help, then please split your oocytes, work with splitting oocytes. Do half of the oocytes with the normal method, and the other half of the oocytes you can try doing it after ethical approval. So yeah, we of course think a little bit different. This paper from Antonio Cabalbo and Laura's group again showed that ionophore, even if the very, very high dosage of 100 micromole and applied for 40 minutes, it's much higher, and the dosage is much higher than whatever is used in IVF. It does not cause chromosome segregation errors. So this is quite reassuring. The gene expression, although it was strontium chloride, is closer to IVF than ICSI without oocyte activation. Markus Montag showed time-lapse data. There is no difference in cleavage rate between normal oocytes and those that had been artificially activated. The language skill, the motor skills, and the cognition up to the age of 10 is completely normal. So the very last 2 slides, and I'm sorry for the delay, is that even in case you use ionophore or ionomycin or electrical activation or whatever you do, you might end up without fertilization. So we do have patients that whatever we try have no fertilization. So you could increase the exposure time, because if you increase the exposure time, you get a higher area under the curve. So there is more calcium is free. The problem is too much of calcium is lethal for the cells. So, and you don't know what is the right calcium level in the cell. But you could switch from 15 minutes, for example, to 30 minutes, or to use a 2-fold exposure like the Belgians do it. Right after ICSI, you have 1 application, 15 minutes, half an hour rest, and then another 15 minutes of ionophore. You could combine those 2 types of ionophore. You could use the very invasive approach, injecting calcium chloride directly, or you could use recombinant PLC zeta, which gives you nice spikes, but it's not available yet, and you don't know the dosage as well. So in case you have no fertilization after ICSI, please do not repeat ICSI, rather go to artificial oocyte activation. And then you could choose between modified ICSI, piezoelectric manipulation, or ionophores. But in case these two methods won't work, you might end up in calcium ionophore treatment anywhere. If this does not work, wait for the recombinant PLC setter, and the last solution, last exit is oocyte donation or sperm donation in case you prove that the sperm is the problem. So last but not least, I'd like to draw your attention to the upcoming Alpha meeting, which will be held in Copenhagen and has a lot of cutting-edge talks, I guess, because the program has been put together by David Gardner. And thank you very much for your kind attention.